Hello, everybody. My name is Omar Farah, and I'm the stem cell field application scientist at Thermo Fisher Scientific. And today we're going to talk a little bit about leveraging our custom cell services to build better models for disease research. So Thermo Fisher has a broad range of extensive service capabilities that spans both model generation as well as discovery and development workflows to help our customers meet their needs. And for this talk specifically, I'll be focusing on service tools we provide for model generation that are outlined in red over here. And these really can be summed up into two categories. The first is a stem cell category. And this category is key for disease model generation. It includes services such as reprogramming, characterization, and cellular differentiation. And then the second category is cell engineering. And this category serves really as a continuum for stem cell services, where our expertise would be leveraged to both design and deliver genomic edits to our stem cells specifically. And it also includes the state-of-the-art genome editing tools such as CRISPRs and TALENT as well. So before I start, I usually like to give a brief introduction to the history of stem cells. And by definition, stem cells have two main properties. They must be self-renewing and give rise to other cell types. And these concepts really aren't all that new, and they can be traced as early back as the 1870s, where Ernest Haeckel used the term stem cell to describe a fertilized egg as the cell origin of all other eggs in an animal or human organism. But fast forward to 1953, which is actually the same year that Watson and Crick published their famous paper about the DNA double helix, two researchers up in Canada published a paper that would first identify the theory of stem cell renewal, and they did so by identifying a population of spermatogonia responsible for the production of spermatozoa in murine models. And so ever since then, growing interest in stem cells and their applications has been on the rise. And more recently, with the work of Shinya Yamanaka and Jamie Thomason in 2006, in the generation of induced pluripotent stem cells, really has allowed for great advances in human-based stem cell models. And so over the past decade or so, Thermo Fisher has created an entire portfolio of products, reagents, and services that support the entire workflow of stem cell culture and disease modeling. And, all the way from the flexibility of model selection to reprogramming, culture of your stem cells, engineering of these cells, as well as characterization, differentiation, and finally functional analysis as well. And so if we start by looking at the cell model stem cell reprogramming services, you'll see that we offer two main methods by which a variety of somatic cells can be reprogrammed into iPSCs. The first is a method that employs the use of episomal Epi5 iPSC reprogramming kit to generate viral-free and transgene-free iPSCs, and that's seen on the top right. The second is an efficient method that employs the Cytotune iPSC 2.0 reprogramming kit, and that kit uses a Sendai virus in order to deliver the reprogramming factors. These methods support the reprogramming of a wide range of cells that include fibroblasts, CD34 positive cells, as well as peripheral blood monocytes or PBMCs. And usually the workflow goes as follows. So somatic cells that you desire to be reprogrammed are thawed and recovered in our facility and are tested for mycoplasma contamination. And as an optional step here, the cells can be characterized and analyzed for any genomic stability or karyotype here. The cells can then be reprogrammed with the desired method that I mentioned. And then following reprogramming of these cells, the pluripotent stem cells that arise will be selected, expanded, and finally characterization of these iPSC lines to assess pluripotency and genomic stability can be performed as well. And so the final resulting output is a generation of donor-specific iPSCs. Now, when reprogramming your somatic cells, like I mentioned, you have two options really that you can choose from, either the Epi5 episomal reprogramming kit or the Cytotune 2.0 Sendai reprogramming kit as well. And so for the episomal Epi5 reprogramming kit, it includes 5 episomal vectors that include the Yamanaka reprogramming factors, the OCT4, SOX2, KLF4, and CMYC, along with a vector for Lin28 and another vector for a dominant negative mutant of P53 to ensure high expression of the other plasmids and increase reprogramming efficiency. And this episomal method is both a non-integrative as well as a viral-free reprogramming method. Now, the Cytotune iPSC 2.0 Sendai reprogramming kit is a method that employs or uses a modified version of the Sendai virus that cannot produce infectious particles, and has really seen one of the highest, or amongst the highest, reprogramming efficiencies in current technologies that are currently available. And the main advantage of using the Cytotune Sendai reprogramming method is that it's non-pathogenic to humans. It does not alter the host chromosomes or genomes, and by that means the non-integrating vectors and transgenes that are used to reprogram are eventually eliminated from the host cell. And then finally, the Sendai virus really has a high transduction efficiency with a low multiplicity of infection. So our customers can also leverage our expertise and experience in genome editing in order to introduce specific mutations into the genome of pluripotent stem cells. Our services offer both CRISPR-Cas9 as well as TALENs as genome editing tools. And such nucleases can be designed to target specific sites in the genome, creating those double-strand breaks at desired locations to introduce multiple modification options that include, knockouts, knock-ins, SNP edits, stop codons, or large donor insertions. And so similar to the reprogramming service, our gene editing service follows a milestone approach. So before starting the workflow, the pluripotent stem cells are transferred and adapted into our facility. This includes the sterility and mycoplasma testing that I talked about, which would not be required if you're already using a Thermo Fisher cell line or continuing service from previous reprogramming with Thermo Fisher. Now, before starting the gene editing workflow, we usually advise to characterize the PSCs in order to ensure their pluripotency and the absence of any genomic anomalies. And we then start by designing and synthesizing the guide RNAs or the TALENs to the specific gene of interest or the loci of interest, followed by the delivery of the gene editing tools, whether it be your TALENs or your CRISPR-Cas9 complexes along with the guide RNAs and the donor DNA. And so after performing the edits, we move to confirm on-target edits, sort and clonally expand the cells, after which we generally recommend to recharacterize the pluripotent stem cell line again in order to ensure that the pluripotency and genomic makeup of the cells was not changed during this process. And this is important because pluripotent stem cells are sensitive in nature, and so stressful conditions and workflows such as gene editing can perhaps introduce undesired changes in the genome. And at the end, what you'll have is your engineered cell line. And so you've heard me talk a little bit about characterizing cells across several stages, whether it be reprogramming or gene editing workflows. And it's really important to characterize your cell lines, especially after stressful interventions, like I mentioned, such as reprogramming, gene editing, or clonal isolation, for example. And we offer a suite of characterization solutions for the qualifications of stem cells. On the left side of the slide, you can see that in order to assess genomic stability, we offer several services, the first of which is a carrier-stat service. And this is an improved alternative to traditional G-banding methods in order to examine chromosomal abnormalities. This service can be coupled with our cell identification service seen on the bottom left to ensure that the cell lines going initially into our pipeline are the same ones that our customers are getting back. The second service that assesses genomic stability is our Oncomine service, and this is a next-generation sequencing-based service designed for monitoring genomic variations associated with select known cancer hotspots in pluripotent stem cells. Now, in order to assess pluripotency of of your lines, we offer the PluriTest, which is a service that utilizes global gene expression in order to position the particular pluripotent stem cell line compared to a database of 264 other established embryonic and pluripotent stem cell lines within our database. And we also offer the TACMAN scorecard service, which uses a gene expression-based assay used to confirm pluripotency. But not only that, it's also a quick and effective method to assess trilineage differentiation potential of your pluripotent stem cell line into the 3 germ layers, those being the endoderm, the mesoderm, and the ectoderm. And the assay consists of a panel of 93 genes that are specifically picked in order to measure pluripotency and germ layer specific bias, it compares the gene expression of those particular genes to 13 established pluripotent stem cell lines in both the undifferentiated and the differentiated states. Now, the stem cell workflow does not usually stop after reprogramming or gene editing. In disease modeling, for example, pluripotent stem cells are differentiated into a specific cell type, where our differentiation services enables the specification of pluripotent stem cells, either with or without genomic edits, to disease-relevant cell types. And here, Thermo Fisher offers various media systems that allow for robust differentiation of PSCs, either into midbrain floor plate progenitors and dopaminergic neurons, neuronal stem cells that can be further differentiated into hindbrain-like neurons, oligodendrocytes, and glial cells. We also offer media systems and kits that support PSC differentiation into definitive endoderm as well as cardiomyocytes. And finally, our custom services can work alongside our customers to generate other cell types, given reasonable feasibility and a published protocol from the literature. I should mention here that although we are able to ship progenitor populations to our customers, we usually are not able to ship terminally differentiated cells. Dopaminergic neurons, for example, do not fare very well once cryopreserved and then rethawed. We thus provide our customers with the floor plate progenitors in that case, along with all the reagents and detailed protocols required in order to complete the maturation process. Of course, we can also terminally differentiate the cells in our facility and run the analysis on the cells, ourselves. And this is actually a great segue for me to talk about our in-house analysis capabilities. So, in very much the same way that that we can help our customers with the differentiation services, we can do the same by supporting our customers' post-differentiation analysis requirements. And this includes, high content analysis such as cell health and morphology assays, neuri outgrowth assays, or culture characterizations. But in addition, we offer services related to functional assays that include assays measuring calcium release or membrane or voltage potential assays as well. And we can also support custom analyses as well with the feasibility of each assay, taking case by case, but this is definitely something that we can have a conversation about with our customers. So what I wanted to do now is just shift gears a little bit and actually give you some great disease modeling success stories that were funneled through collaborations between Thermo Fisher and the Thermo Fisher custom services and our customers. And the first is a collaboration with the Parkinson's Institute to generate a Parkinson's disease model. Here, a stable Cas9 expressing iPSC line was engineered to introduce a point mutation in the LRRK2 gene, specifically a G2019S SNP. This particular SNP is associated with Parkinson's disease, and these iPSCs were then characterized and differentiated into dopaminergic neurons using our dopaminergic neuron differentiation kit. And then a number of assays were then used to look for phenotypes in those neurons to check whether the introduced mutation had any effect on the biology or the function of these neurons. Here's a little bit more information about that iPSC line that I just talked about. So here, a Cas9 vector was stably integrated into our GIPCO human episomal iPSC line via lentiviral delivery. And the selection of these cells was subsequently done with antibiotics. The resulting iPSC line was the one that I mentioned on the slide before, and that is an iPSC line that stably expressed the Cas9 protein and would only need the delivery of guide RNAs and or donor DNA to edit a specific part of the genome. And you can see how this Cas9 iPSC line can serve as a great tool for easy generation of human-induced pluripotent stem cell-based disease models. And in true form to our workflows that I've discussed earlier, this particular cell line, after it was generated, was characterized and assayed for its ability to differentiate into the 3 different germ layers, and you can see that in the bottom panels over here. So after introducing the LRRK2 G2019S SNP, we reconfirmed targeting of the LRRK2 gene by Sanger and next-generation sequencing, seen in the bottom left panel. We then examined the quality of the edited iPSCs. We ran the TACMAN scorecard, the pluritest, and the karyostat assays to assess the state of pluripotency, as well as the karyotype of the edited iPSCs. And you can see that the edited iPSCs are shown to be pluripotent by both the scorecard and the pluritest assay. In the pluritest assay, seen in the top right panel, you can see that the LRRK2 G2019S mutant clusters with other validated iPSC lines, which is the red cloud. In addition to the LRRK2 G2019S, the LRRK2 G2019S mutant displayed a normal karyotype as judged by the karyostat assay. And this is seen on the bottom right of your slide. And just for a little help in interpreting this karyostat figure, you can see that the x-axis represents the chromosome number, while the y-axis represents copy number. And here you can see that the mutant line that we created displays a copy number of 2 for all chromosomes, with the exception of the Y chromosome. And this was expected because the parental line that we used was derived from a female donor. And so after making the edits, both the control and mutant lines were differentiated using our dopaminergic neuron differentiation kits, and both lines differentiated normally. Tyrosine hydroxylase, which is a dopaminergic neuron marker, and beta-3 tubulin, which is a general neuron marker, appear to be comparable in both the mutant line seen in the top panels as well as the control lines seen in the bottom panels. So things look pretty similar for both the parental and mutated cells at this point, really proving that the mutant line can be differentiated into dopaminergic neurons. Now, in order to analyze neuron biology and function, we looked at neurite outgrowth. We looked at this by using a live cell imaging system and found a slower rate of neurite outgrowth in early time points for the G2019S mutation. However, after a longer period of time when neurite outgrowth stabilized, those mutant cells appeared to actually have longer neurites than in the control parental lines, or compared to an SNC A30P either heterozygous or homozygous mutant. And just to clarify here, an SNCA is another gene that's associated with Parkinson's disease. So this is a line that we're using as an additional control. So this particular phenotype that we saw as far as neurite outgrowth was specific to the G2019S mutation in the LRRK2 gene. Now, one of the hallmarks of Parkinson's disease is the loss of dopaminergic neurons. So in order to model the disease, we looked at apoptosis and sensitivity of the differentiated cells to toxins and neurotoxins. And 6-hydroxydopamine is one such neurotoxin that's specifically uptaken by dopaminergic neurons. Now, using our Cas-based 3/7 high-content imaging assay, we asked how the control or mutant dopaminergic neurons responded to this neurotoxin. And in this experiment, stratosporin, which is a potent non-selective apoptotic agent, was also used to set a reference baseline for overall cell death in both the control and mutant lines. And on the left side of the slide, you can see some of the representative images from the high-content imaging assay. Now, on the right side, the top right panel, you'll see the quantified normalized data from the imaging assay, which might be a little bit easier to interpret. For both the DA neurons in the first column of our Cas9 parental line, our LRRK2 G2019S homozygous mutant in the second column, and then our SNCA A30P heterozygous in the third column, and SNCA A30P homozygous mutant line on the last column on the right. And as you can see, the LRRK2 G2019S mutant, again in the second column, is much more sensitive to the exposure of 6-hydroxydopamine, showing increased apoptosis. Now, in order to mimic stressful conditions, we repeated this experiment while culturing the control and mutant lines in the absence of glucose. And as seen on the bottom right graph, you can see that the mutant line showed increased apoptosis compared to the parental lines. The SNCA homozygous mutant DA neurons also showed a higher degree of apoptosis under stressful conditions. And some of this data was previously published by other groups. So it's nice to see as a proof of concept that our assays are also able to replicate previously published data. Now, finally, it's really known that the LRRK2 G2019S mutation can have an impact on gene expression of a number of genes in dopaminergic neurons. So we looked at some of these genes using the TACMAN gene expression assays. We saw that by introducing this particular mutation, an increase in the expression of LRRK2 regulated genes was observed in the mutant line compared to the parental line, and this is easily visible in the bar graph on the left. Now, interestingly, no changes in gene expression was observed in the SNCA mutant line. And this, again, is consistent with some of the published data in the literature, but what it strongly suggests is that these changes in expression patterns are specific to the LRRK2 G2019S mutation. And so, to summarize, we were able to engineer the LRRK2 G2019S mutation by utilizing our Cas9 expressing iPSC line. We confirmed that these edited iPSCs maintained their pluripotency and had a normal karyotype. And then we were able to successfully differentiate the edited iPSC line into midbrain dopaminergic neurons and discovered that the specific mutation affected neurite outgrowth, and neurotoxin sensitivity, as well as specific gene expression patterns in those cells. Now, another disease modeling success story that I wanted to highlight is a case where you use a very similar workflow to generate another disease model, but for dilated cardiomyopathy. Here, the same Cas9 expressing iPSC parental line was used in order to generate a mutation in the troponin 2 or TNNT2 gene, specifically an R141W SNP mutation. Again, just like before, this line was characterized and then differentiated into cardiomyocytes here using our cardiomyocyte differentiation kit. And then several assays were performed in order to understand the effect of this mutation on cardiomyocyte function. Just like before, after introducing the R141W mutation into the troponin 2 gene, we've assayed for a pluripotency and karyotype, and we saw that the edited line displayed normal karyotype as well as pluripotency, just like we did before. After confirming the genomic edits, both the Cas9 parental control line and mutant line were differentiated into cardiomyocytes. And here you can see that the mutant line differentiated normally similar to the Cas9 parental line. And this is really evident as you see both the control and mutant lines saying positive for TNNT2 in red or actin-1 in green. And these two are structural proteins that are essential for the contractile apparatus in cardiomyocytes. Again, indicating here that cardiomyocytes were successfully derived from both the TNNT2 R141W mutant line as well as our parental control Cas9 iPSC line. So we then examined whether functionality in the mutant cardiomyocytes is compromised by monitoring calcium transients. The calcium transients in the cytosol controls each cycle of contraction and relaxation in beating cardiomyocytes. And so using FLO4, which is a dye that allows for the visualization of calcium levels inside the cell, a clear delay in calcium decay is observed in the mutant cardiomyocytes compared to the control. And you can see this in the top left graph. Now, this delay in calcium transients can be correlated with slightly lower beat rate observed in the R141W mutant cardiomyocytes compared to the wild type, which again you can see in the top right panel as well. Now, Under stressful, or I should say, usually under stressful conditions, like extensive exercise, for example, cardiomyocytes increase their base rate to support the increased blood flow. And cultured cardiomyocytes can be stimulated with isopropanol to accelerate their beat rate, whereas DMSO administration does not do that. And this is often used as a model to mimic cardiac stress in vitro. And you can see this in the bottom left panels. Now, in the case of the TNNT2 mutant, a spontaneous contraction is rapidly compromised after isopropanol administration. And although no published data is available for this particular SNP, the R141W, as it relates to cardiomyopathy in humans, cardiomyocytes carrying a similar SNP on the same gene show similar phenotypes. And so here, to summarize, again, we were able to generate a TNNT2 R141W mutation by utilizing our Cas9 expressing iPSC line again. We confirmed that the edit was there, and that these cells maintained their pluripotent status as well as maintaining normal karyotype. And in the process, we were able to discover that this specific mutation affects spontaneous contraction and showed cardiac arrest after being challenged. Now, the final case study that I wanted to talk about is a really interesting story that talks about the generation of hypoimmunogenic derivatives of pluripotent stem cells that can evade immune rejection upon transplantation into immunocompetent recipients. And this work was done in collaboration with Dr. Sonia Schreffer's group at UCSF. So during pregnancy, the maternal immune system is tolerant to allogenic paternal antigens, although it would reject similar cells from the same baby later in life. And after examining gene expression patterns of syncytial trophoblasts, which are the fetal placental cells that are directly at the maternal-fetal interface, Dr. Schreffer's group postulated that hypoimmunogenic iPSCs can be generated through the mimicking of syncytial trophoblast gene expression patterns, which were mainly accomplished by inactivating MHC class 1 and class 2 genes, as well as the overexpression of CD47. So in order to test this hypothesis, MHC class 1 in genes were targeted by knocking out beta microglobulin 2 and C2TA genes, followed by overexpression of CD47 in our Cas9 human iPSC line. The iPSCs were then differentiated into various cell types, including cardiomyocytes or endothelial cells, and finally these cells were transplanted into recipient mice in order to assess their survival and immune interactions with the host. So although Dr. Schreffer's group was able to recapitulate their finding both in mouse and human iPSCs, I'll mainly discuss the generation of the human hypoimmunogenic iPSC line. And first, both the human beta-2 microglobulin as well as the human C2TA genes were simultaneously targeted for CRISPR-Cas9-mediated destruction. Secondly, these edited iPSCs were transduced with a lentivirus carrying human CD47 complementary DNA downstream of an EFS promoter to ensure its overexpression. So Dr. Schreffer's group subsequently differentiated these engineered iPSCs into cardiomyocytes and endothelial cells. And the cell lines were transduced to also express a firefly luciferase reporter in order to enable tracking by bioluminescence imaging. And transplantation studies of these engineered grafts were then conducted in humanized allogenic mouse models. And interestingly, both the engineered iPSCs and the engineered iPSC derivatives, whether it be cardiomyocytes or endothelial cells, evaded immune rejection when grafted into the humanized mouse models. Now, only the engineered cells survived long terms without, and this was without the use, without the use of immunosuppression in these mice. This was not the case for wild-type iPSCs or wild-type iPSC derivatives, where you can see that they were clearly cleared out by the immune system as represented by the gray bars. And you can imagine the magnitude and an impact this kind of work will have on universal transplantation therapies. And it is really nice for us to say that we were able to contribute to this work in order to, leveraging our technologies and our expertise in CRISPR-Cas9 and lentiviral-based gene editing tools in order to make this come to fruition. And then finally, on this note, I'd like to leave you with this slide that, again, really highlights the wide range of expertise and services, that Thermo Fisher has that spans over the entire stem cell and disease modeling workflows. And again, all the way from the flexibility of model selection to reprogramming, culturing of the cells or engineering them, as well as characterization, differentiation tools, and finally, even functional analysis as well. And with that, I'd like to thank you very much.